All right, hi everyone. Welcome to today's virtual learning experience. In this session, we'll be introducing you to the wonderful world of Bioconductor. Bioconductor, in case you don't know, really is a one-stop shop for all things computational biology. I hope that by the end of this session that you will love this resource as much as I do. So we're going to begin today's session. Oh, I should say that throughout the session, please feel free to enter any questions you have in the little box to the left of me, and I'll do my best to answer them live. So let's begin by taking a brief tour of the Bioconductor website. So you can see here we're on the homepage of Bioconductor. Bioconductor is many things to many people. So to some people, Bioconductor is a repository of computational biology packages. So we can see here that it currently has 2,230 software packages. I should say that Bioconductor, these packages are packages that are run in the programming language R. So you do need to have R to be able to make use of Bioconductor. If we click on this and take a look at some of these packages, we can see that there are clearly many packages. And these packages allow you to, for example, perform differential expression analysis. They allow you to visualize your data. There are different packages for different kinds of omics analysis. For example, if I'm interested in, let's say, expression analysis, if I just type in expression, I can see some really well-known packages in the field that are used for performing expression analysis, like EDGAR and DESeq2. In addition to being this wonderful repository of these amazing packages, Bioconductor also is this excellent resource of educational material. So if we click on, for example, education and training, we can see that they have these different modules that they're developing and have developed to help people of all competencies and experience levels learn. So they have this general introduction to data science, a more thorough introduction to the Bioconductor project, and this introduction to analyzing bulk RNA-seq data. They have some book recommendations, all of which, or most of which I can attest to are really good resources. There's a fantastic support site. So this is where you ask if you have any questions about an analysis that isn't working so well, this is the place to come. One of the great features about this particular forum is that if, for example, you have a problem to do specifically with the DESeq2 package, and if you enter a question about there's a very good chance that the creator of DESeq2, Michael Love, will answer you. I mean, this is pretty fantastic. Like here, someone has a question about EDGAR heat map construction. Gordon Smith, who, if you don't know who he is one of the biggest names in computational biology. And the fact that he is sitting here answering questions from strangers is truly remarkable. So that's really all there is to say about Bioconductor. It's you can go down various rabbit holes. There's the package rabbit hole, there's the education rabbit hole, there's a forum rabbit hole. I encourage you to explore every aspect of this wonderful resource. One thing I use Bioconductor a lot for is to learn about new packages. So I mentioned this package DESeq2, for example. So let's say you're not very familiar with this package and you want to learn about it. The easiest thing to do is, or an easy thing to do, is to enter the name of the package into the search bar. We hit enter. We can see we have quite a few hits. Generally, the top hit's the most useful one. And so this is an example of a page that's associated with a particular package. So the name of the package is DESeq2. We can see that it has a title, Differential Gene Expression Analysis Based on the Negative Binomial Distribution. It sounds a little bit complicated. It is a little bit complicated, but all this means is it allows you to perform differential gene expression analysis using RNA-seq count data. And because count data have different statistical properties compared to continuous data, we need this, or not need, but negative binomial regression is a preferred approach to analyzing those kinds of data. It's associated with a paper. So this was a paper that was published in 2014 in the journal Genome Biology. I should say that even though in this specific instance, the paper was published in 2014, this package is under continuous development. It's just that the details of the statistical methods that are used in this package are most thoroughly described in this paper here. We can see that there are some pretty simple installation guidelines. So the Bioconductor, the main Bioconductor package is this package called BioC Manager. And so if you run these 3 lines in R, the first thing it'll do is it'll look to see if you have the BioC Manager package installed. If it detects that you don't, it'll install that package, and then it'll use the install function within that package to install DESeq2. So it's generally pretty pain-free to install Bioconductor packages. Sometimes there are issues, but they can be usually pretty easily fixed. The last thing I want to highlight on this page is this vignette section. Different packages have different qualities of vignettes. I mean, some packages don't even have vignettes. All they have is a kind of difficult to read manual. But many packages, especially the more popular ones, tend to have excellent vignettes. So what's a vignette? If I click on this HTML link, it takes me, actually I can close the Bioconductor website. It takes me to the vignette that these authors, Michael Love, Simon Anders, and Wolfgang Huber, again, if you aren't familiar with these 3 people, giants in the field. They have very recently updated this vignette. This is just a few months ago. And it's analyzing RNA-seq data with DESeq2. And you can see here that this is a book-length vignette. This really will have 95% of everything you would want to know about RNA-seq analysis using this package. I frequently use these kinds of vignettes as references, so sometimes I'll forget some detail about some aspect of, for example, differential expression analysis, so I can just jump to that bit. And what's really nice about these vignettes is that they are typically entirely reproducible. So you can see that the data that they're using here is data that you can use too to reproduce everything you see here. This one in particular is really well documented. And you can see they show you how to use the different functions, what they mean. They discuss the outputs. They discuss some of the statistical details. It's really, really fantastic. And then if I, like, for example, I forget the details or the nuances of producing a heat map using count data, I'll click on heat map of count matrix and I see there's a code, there's an example output, what it means, and so on. So this is an example of a really, really good vignette. And I'll be putting links to other similar vignettes that I think are really useful. So in the description for this VLE, I put in a link to this paper here, which is now a landmark paper called Orchestrating High-Throughput Genomic Analysis with Bioconductor. And so this was published in 2015. So the Bioconductor project has been going on for a while now. Speaking of giants in the field, so there are many, many authors on this paper. Every single name here will take you down another rabbit hole of some important aspect of computational biology. I encourage you to find out more about each of these authors. So this is a really nice paper that introduces you to the philosophy of the Bioconductor project, and the authors here describe their motivations for creating and maintaining the Bioconductor project, which I will not try to summarize here. I do recommend that you read this paper. It's really nice. It's not particularly long. What I do want to highlight is in this paper is this figure 2, which I appreciate might be a little bit difficult to see. So what I'm going to do is hop on to another vignette. And this is a vignette on this object called the summarized experiment object. So what we can see here is this figure in the middle of my screen, which might look a little bit complicated. The orchestrating high throughput, what's the title? Yeah, high throughput genomic analysis bioconductor paper. One of the things that it introduces readers to is this concept of an S4 object, which this is a depiction of the architecture of this object. An S4 object is a particular type of object in R. So R has various kinds of objects, vectors, matrices, data frames, lists, and so on. The S4 object is an exotic object type that I have not used in any other context besides the kind of bioinformatics analysis I do. Uh, and this particular type of object offers a unified, um, common way of handling different kinds of omics data. So whether you're working with single cell RNA-seq data or proteomics data or bulk RNA-seq data or methylation data, there's typically some kind of S4 object that allows you to handle these complex data in a unified and systematic fashion. So that might not make a huge amount of sense. So what I'm going to dedicate the rest of this talk to is an exploration of some of these objects, which I hope will give you a better sense of what these objects are and why the Bioconductor project people have chosen to use this particular object type. And I really hope that by the end of this talk, you will um, appreciate uh, why this is such a, or can be such a useful object. So let's begin with a description of the architecture of this object. So let's start in the middle. So you can see in the middle, you have these rectangular sheets. Think of each sheet. So let's look at this blue one, looking at this blue one here. Think of that as just a gene expression, a table that contains gene expression data. So you have rows that correspond to individual genes, and you have columns that correspond to individual samples. And within this rectangular object, you have gene expression values. So you can think of this as a data frame, a matrix, or like an Excel sheet. But you can see that there's more than one. There are, in this case, 3. So the way to think about this is kind of like in Excel or Google Sheets. You have sheet 1, sheet 2, sheet 3. You can similarly have sheet 1, sheet 2, sheet 3. The difference is in this particular object type, these multiple sheets are connected to each other. They're communicating with each other in a way that Excel sheets are not. What do I mean by that? In the case of Excel, if you make a change on sheet number 1, that change isn't going to automatically be detected in sheet 2 or sheet 3. But here, the amazing thing about this object is every part of it is communicating with every other part. So you have these, so this central portion here, this is the meat of the object. This is the portion of the object that contains um, all the measurement values. So in this case, gene expression values. You see this rectangular object, this narrower rectangular object on the left here. This contains metadata that are associated with the rows of this large rectangular object. So in this case, that would be the genes. So this rectangular object, maybe the rows, they just have ensemble gene IDs. But there's a lot more to a gene than just its gene symbol. You might want to know something about its function. You might want to know how long it is. All of that information can be stored in the metadata for the rows, in this case, the genes. And similarly, you will likely have metadata associated with the samples. And that can be stored in this rectangular object at the top right-hand corner. So if your samples, let's say you were working on mice, the metadata could include information about the sex of the mice, the age, whether they were treated with a particular drug or not, whether they were wild type or knockout, and so on. So this is a way of bundling together data and metadata into one object in a way that connects all of these different parts together. Because if we didn't do it this way, the alternative would be to have, for example, your sample metadata in one data frame, your array data in a matrix, your log normalized array data in another matrix, your gene metadata in another data frame. And things can get out of hand really quickly. In addition to all of these things, you also have just general kinds of metadata, like when was the experiment done? What kind of experiment was done? Is this associated with a publication? And so on. Um, so this is a very like high level overview of this S4 object type. And if this is your first time thinking about this kind of object, this picture here will likely not make a huge amount of sense. However, when you start to play around with these objects, I strongly encourage you to keep revisiting this image, because I promise you this will help. This will help produce a mental model of this object type that is really, really useful. The last thing I'm going to say about this image before switching over to the code is that underneath each of these shapes, you can see that there are some functions. And these are the functions that we will be running in a minute, and we'll see what these different functions do. And what's amazing about these functions is that whether you are dealing with microarray data or RNA-seq data or single-cell RNA-seq, a lot of these functions will stay the same. So it's not like you have to learn this completely new way, this completely new set of functions when you're handling one type of omics data and another type of omics data, which I think is a really powerful feature of this S4 object class system. So if we switch over to our studio, we can see here that I've made an R Markdown file to go with today's learning experience. So this is in right now stored in a GitHub repository, and the link to that GitHub repo is in the description on YouTube, so underneath me right now. And so you are welcome to clone that repo, fork that repo. You can just download this RMD, do whatever you want. Um, I'll give you the option to either sort of kind of code along with me if you'd like, or you're welcome to. Actually, I mildly, slightly prefer just watching this. And then if you're really interested in the code, I encourage you to go through the code by yourself afterwards. So let's begin by first loading a few necessary packages. So you can see here, I've commented out the installation instructions because I already have these packages. But of course, if you don't have any of these packages or you don't have some of them, please uncomment the relevant lines and then run through everything. So we'll be loading the tidyverse, which has some useful functions. And we'll be loading some of these packages. So you can see we have the summarized experiment package. So this is the one. So the summarized experiment, a summarized experiment object is an example of this S4 object type I've been talking about. Airway and ALL are two packages that just contain data. So Airway contains RNA-seq data for a particular experiment, and ALL contains microarray for another experiment. And then we have the Limma package, because I'll be ending today's learning experience with a kind of sort of end-to-end analysis where I start with a summarized experiment object, and then I'll show you how you can use that object to perform differential expression analysis. And then finally, we're going to briefly use this Biomat package. The Biomat is a really, really great package for translating from like one. So, for example, let's say you have on gene entree IDs and you want to convert them to gene names. Biomat is the package for you. And importantly, except for the tidyverse, all these five packages here, they're all from Bioconductor. So they hopefully will give you a sense of the variety of useful packages that are in this repository. Okay, so let's begin. So before we get to the code, I just want to point out that I've put links to to some of the things that I've mentioned so far. So the orchestrating high throughput genomic analysis with bioconductor paper, there's a link here. And then the image of that S4 class object that I showed you, that is taken from this vignette. So you can click on it. I strongly recommend that you go through that vignette. It's really well done. So beginning with the code, let's first load this airway object. And before we take a look at the object itself, let's first just quickly look to see what the data, where the data come from. So what I did so I'll be using the help section a lot today. I use the help section constantly because there's a lot to remember. And thankfully, the help sections for a lot of these packages and a lot of the functions are written really well. So to pull up the help page for really anything in R, you first put question mark, and then you write the name of the thing you want to get help on. So in this case, on line 39, I just put question mark airway, and it opens up this help page, and it says, it has a description. It's read counts. So I know it's RNA-seq data. Read counts per gene for airway smooth muscle cell lines RNA-seq experiment. Great. And actually, if I scroll all the way down, these data were taken from this published paper. So if I'm interested in the details, I can go to that paper. But that's really all we need to know. We don't, for this part of the demonstration, we won't really be discussing the experimental details. We'll just be playing with this object. So because I don't want to keep typing airway, I'm going to assign airway to an object called SE. SE stands for summarized experiment. And the first thing I'm going to do is just take a look at SE. So if you look at my console here, I'm just going to make it a little bit bigger. You can see that it's class ranged summarized experiment. So again, if this is your first time being introduced to these kinds of objects, you will be introduced to various potentially confusing names. There are summarized experiments, there are ranged summarized experiments, there are Q features, there are single cell experiments. So these are all different names of the same type of object that are used for different purposes. So a ranged summarized experiment object is typically used for RNA-seq data and microarray data. So that's the class. But beyond that, what are the details of the summarized experiment object? The names, don't worry too much about it. Let's look at what comes after this. So we can see that there's some useful information just in this these few lines. We can see dim. It tells you the dimensions of this object. So there are 63,677 rows. So in this case, that means there are that many genes, and there are 8 columns. So in this case, 8 samples. There's a metadata section. There's an assay section. So the assay section is where you have the actual count data. And then you have row names, and this just shows you a few of these row names. So these are the ensemble gene IDs. And you have row data, which I will get into in a second. You have column names. So these are the names of the columns, which are the samples. So you can see a few of them. They have weird names like SRR, something, something. And then you have this thing called call data, which I will get into a second also. All right, so this, if you just execute SE and you see this sort of gives you a very, very, very high level overview of what this object is and what it might contain. But we don't really see very much that's immediately useful here. So arguably the most important thing in that this object contains is the sheet that contains the count data. So how do we obtain this count data? So once again, this assays section of this object, you can see here it has it says assays and then in parentheses just the number 1. That means there's just one assay. What's amazing about these objects is you can have as many assays as you want. In this case, there's just one, and it's called counts. So to visualize, to access that count data, you can use the assay function. Before running the assay function, I'm just going to do $assay. You can see it takes me to this help section here. This help page doesn't just provide you information about this assay function, which is technically called an accessor. It also gives you information about these various accessors that can be used for these sorts of objects. So there's a lot here. I'm just going to put this up here as a reference for myself, but it's a little bit difficult to go through this. It's easier to go through this whole help section by yourself. So the assay function works it's pretty easy to use it. You just do assay, and then you give the name of the object. So I've called it SE. And actually, if you just run it like this, it works. And if we look at our console, we can see what look like count data. You can see the rows are all genes, and the columns are our samples. A better way of accessing the count data is to explicitly give the name of the assay as an argument. And the reason you do that, in this case, it's not necessary because there's only one assay. But if there were more than one assay, this would be a better way of pulling up the relevant data. There's another way of doing this that I'll just show. We can use this assays function. In this case, we do assays, the name of the summarized experiment object, and then $, we give the name of the assay, $counts. It gives us the same output. So far, so simple. So this is how we go from this SE object to obtaining the count data from this SE object. Okay, so the next question is how many rows and columns do we have? We can use this dim function here. It's a pretty simple function. Actually, let me clear my console and run that again. You can see it just gives us the dimensions of the assay. 63,000 something rows, 8 columns. Pretty simple. View information related to genes, if any. So by this, I mean, let's see if there are any metadata that are associated with our rows, so with our genes. And to do that, we can use this function called row data with a capital D. So if we do row data SE and run that, we see we get this. This might be a little bit harder to read, but once again, the output contains rows that correspond to genes, but now the columns aren't samples. The columns, each column contains some information about that gene. So we have 10 columns, as it says here. There's a gene ID, which seems redundant, the gene name, entree ID, what's the gene biotype, is it protein coding, is it a link RNA, and so on, gene start, gene sequence start, gene sequence end, and so on. So this is the part of this summarized experiment object where you can start to put in this sort of useful metadata. Because, for example, like these ENSG IDs, they're really hard to read and remember. If we didn't have this object type, there's a good chance that you would have another data set where you would have a list of these genes along with, for example, their symbols. But this way, you can have this useful gene metadata information bundled together with your experimental data, with your count data. And I sometimes refer to these summarized experiment, these S4 objects, as briefcases. And so just like a briefcase can contain many things, these summarized experiment, these S4 objects can also contain many things. So that's how you view information related to your rows, which in this case are genes. So how do you view information related to your samples? We can use this function called call data, C-O-L capital D. And we see, so this is where it gets a little bit tricky. Actually, let me just first, let me rerun the assay just to remind you, when we run the assay function, we see that the output columns are the samples. However, when we run call data, the output rows are the samples. So think of it as you're sort of transposing, it's sort of a transposed version of the assay data. But however, the rows here are the samples. The columns now contain the metadata associated with the samples. So we have the sample name, what cell type was used in this particular case, this dexamethasone, was it treated, was it not treated, and some other information. And so here we can see, for example, let's say in addition to this, let's say this were some kind of clinical study, and you had, so here the samples are now patients, and you had, for example, blood glucose levels, measurements that you wanted to include in some kind of linear regression model. You could have a separate data set with all of this clinical information, or you could just as easily put all of that relevant clinical information into this part of the summarized experiment object, right? You would just have another column called blood glucose, and it would have the readings here. So this way, everything belonging to the same project or the same experiment stays in the same place. So that is how call data works. Okay, moving on to the next bit. I promise that in a few seconds, or maybe not, maybe in a couple of minutes, I'll take a look to see if there are any questions. But moving on, how many treatment groups do we have? Okay, so if we rerun call data, what I mean by that is by treatment group, I'm referring to this, it's not particularly clear there, sorry, but I'm referring to this column called dex. The treatment here is dexamethasone. So so within dexamethasone, how many groups do we have? So just by eye, I can tell that there are only two groups, untreated or treated. But let's say let's say there were, 2,000 samples instead of 10 or however many samples there are here. How would I get this information? Well, one way is to just so we're actually two ways. You can do call data, SE, and then using the dollar symbol, you can extract the dexamethasone column. If you do that, you see here, this is what's known as a factor, and a factor has levels. And this factor has two levels, treated and untreated. So I know there are two groups, treated or untreated. The alternative way of running this actually is just to do SE $DEX. This might seem a little bit strange, like this is how you would pull out a column from a data frame, but SE is not a data frame. It's this exotic S4 object. But in many ways, you can kind of treat it like a data frame. That might be a little bit confusing. I apologize for that, but that's just the way this is constructed. My point is, whether you run line 64 or 65, you still get the same output. And the final accessor function I want to introduce you to is a simple metadata function. So you just do metadata and the name of the object, and you can see that in this case, the metadata that's included contain information about the title of the study, the PubMed IDs. Apparently, there's an abstract. Not especially useful now, but in the second example that we will take a look at, the metadata function will come in handy. All right. So these are a few simple functions that we can use to access different parts, data related to different parts of this SE object. Now let's look to see how we can do some subsetting. So just like you can do subsetting on a matrix or a data frame, you can also do subsetting on these summarized experiment objects. So to warm up, so on line 72, it says, let's subset this so that we can only view the first 5 samples. So I mentioned that we can often treat this summarized experiment object like a two-dimensional object, so like a data frame. And so if SE were in fact a data frame and we wanted the first 5 samples, meaning the first 5 columns, we would just do SE, open close square brackets, space, comma, 1 to 5. And you can do the same thing with this object. The only difference is if you run this like this, so I'm not saving this object, just yet. I just want to see the output. You see, whenever you run a summarized experiment object by itself, you always get this very high level view of the object and what it contains. If I actually want to see the assay itself post subsetting, I can pipe this into the assay function, right? And now you can see, so where previously we saw more than 5 samples, now you only see the first 5 samples. And of course, you see all the genes because we haven't done any subsetting by row. Why did I do pipe? I use pipes a lot. The alternative is to just do assay, SE, and then you subset 1 to 5, and you get the same output. All right, so let's look at the first 5 samples. Let's look at now just the first 5 genes. Simple, you just do SE 1 to 10, and then comma, right? I'm not going to look at the assay now, but because I've shown you how to do it previously. But you can see here, if you look at the dimensions of this object, now instead of 60-something thousand rows, you only have 10 rows because you've only chosen the first 10 genes. All right, what if we want to subset both? Again, very easy. First 10 rows and first 5 columns. So we get this very, very small object. It's now asking us to save this. So we'll save it as SE subset. Now let's view the count data from this subset. That's easy. We've already seen how to do that. We just do assay. Okay, my console. And now when we look at the assay, it all fits on our screen because we can now have an object that only contains information about the first 10 genes and the first 5 samples. And let's view, so this is where things, for me at least, get interesting. So this is the assay data. All we've done so far, we've done the subsetting on our assay. But what does our sample metadata look like? Has that changed? Let's see. So to see the sample metadata for this SE underscore subset object, we do this call data SE subset. Let me clear my console. Now when we run this, notice that the sample metadata have also been updated, right? Previously, we had sample metadata to do with all the samples that came with this SE object. I think it's 10 or whatever. But now we did the subsetting so that we would only get the first 5 samples. Even though we didn't explicitly do anything to our sample metadata, if we look at our sample metadata, we can see that it has tracked what we've done to our assay. And this is remarkable. So if the alternative to this would be, for example, if you had your assay data in one data frame and your sample metadata in another data frame, anything you do to the first data frame is not going to be reflected in the second data frame, as in any changes you make. Similarly, if you had your assay data on sheet 1 of an Excel sheet and your sample metadata on sheet 2, you remove the first 5 columns, or you only keep the first 5 columns for the Excel sheet 1. That change will not be reflected automatically in Excel sheet 2. So this is a very, very powerful way of keeping things synchronized. All right, then I say subset for only those samples treated with dexamethasone. So this is where the subsetting can get a little bit fancy. So it's on the sample. So we'll do SE, and we can do SE $ dex equal to TRT, right? Because those are the only ones that have been treated. And if this, actually, let me clear my console and run that again. If this kind of notation seems a little bit strange to you, this is the same sort of approach you would use for subsetting a matrix or a data frame. So the reason I'm showing this example here is to show you that there's a consistency between the approaches that you would use on a traditional two-dimensional object in R and a summarized experiment object. So I write here, notice how the various parts of this object are connected. So similarly, if I go back up and let's say I pipe that into call data, right? Actually, let me clear my console, run that again. Now, when we look at the dexamethasone column, we only see the treated samples. So again, these different parts of the summarized experiment object are all talking to each other. Pause for questions, delete me. All right, I'm now going to take a quick look to see if there are any questions. No questions? Fine. Okay, so moving back. So these are ways of accessing information from the summarized experiment object, and I've shown you also how to do some simple subsetting. Let's now see how we can modify the summarized experiment object. So let's add today's date as a new column. So to add, so actually I haven't specified here, but I can, I think a good place to put today's date would be the sample metadata section. So the way to do that is to, again, use this call data function, which we have so far only used to access information, the sample metadata information. We can also use the same function to update or change that section of the summarized experiment object. So to do that, we do call data SE and then $, and I can do something like date, right? I'll give it today's date. So 10/18/2023. We run that. So let's take a look at that summarized experiment object now. Just run SE. Do that again. We can see it's slightly hard to tell, but if you look at call data, the very last thing is date. So a new column has been added. And to see it more clearly, if we run call data on SE, we now see the sample metadata now contains a new column called date with today's date. Okay, let me put that here. That's a way of updating, in this case, the sample metadata. How do we remove this column? Easy. Again, you do call data, SE, $date, and then you assign this null object to this column. Now, when we run it again, call data SE, we can confirm that we've removed our date column. So let's look at a more meaningful way of updating our metadata. So it says here, compute mean gene counts and add this information to SE, right? So what I mean by this is the mean gene counts, I mean, taking all the raw count data for a given gene across, what is the mean of those counts across all the samples? So let's break this down. So to compute the mean counts for each row, you can use this really handy function called row means. And we pull out the count assay from this SE object by doing assay SE comma counts. And if we just run that, we now see in what you see in the console is a named numeric vector. And this is a vector of all the genes, the mean count data for all the genes. So that's just computing the mean count data. How do we do the next step of adding this information back to the SE object? That's also easy. What we can do is, so let's add this to our row metadata section, right? Because that's the section that contains information about our genes. So to do that, we just do row data SE, and once again, $, because we're creating a new column, we can call it mean counts. If we run this, our object has been updated. And to confirm that it's been updated, let's once again run row data on the SE object. Okay. You see here, where there were previously 10 columns, there are now 11. And the 11th column has our mean count information, right? That contains the mean counts for each gene across all our samples. We can now use this computed mean count data to do something useful. So on line 114, it says, use this information to filter out genes that aren't detected in any sample. And this seems like a sensible thing to do because if your gene is not expressed at all in any sample, it's probably not a very interesting gene. You might as well just get rid of it. So how do we do this subsetting? Easy. You do SE. You can again use the square brackets. And this is now on the rows. We do This would be for each row, so we can do row data SE $ mean counts greater than 0. Right? And when we run this, might be a little bit hard to tell, but if we look at the dimensions of this object that we've just produced, notice that now there are only 33,000 and something rows compared to the original 63,000 and something rows. So we can see there's like 30,000 genes that aren't expressed at all. And we've just, in these two steps, gotten rid of those genes. Final thing I want to do is to show you, I mentioned that these objects, these summarized experiment objects can contain multiple assays. Right now, the summarized experiment object we have contains only one assay. So let us now log transform our count data, which is a pretty common step. Let's log transform this, but let's also keep our original raw count data, right? Because it's always a good idea to keep your original data pristine. So how do we do that? Firstly, how do we log transform our data? Easy. Let's log base 2 transform. So we do log 2 and then assay se comma counts. And let's just run this as is, and you can see that this looks like log transform data. And the reason I know that is, well, for one thing, the numbers are smaller. And two, you see these like minus infinity symbols. It just means that it's tried to compute the log of 0, which is not possible. So we could, this is why some people add like a pseudo count to avoid this minus inf, but we're not doing any analysis on this, so we can skip that for now. So that's how you log transform, but how do you create a new assay? So this is where the assays function can be used. So we do assays, SE, $, and we give our new assay a name. Log counts seems reasonable. And then we assign the log transform data to this new assay. And how do we see this assay? Well, for one thing, let's just run SE again. Look at the output. Now, when you look at the assays section, instead of 1 in the parentheses, you now see 2. And you see that there are 2 named assays, counts and log counts. And if you want to see log counts again, extracted from this SE object, easy. You do assay, SE, and now instead of putting in counts, you put log counts. We run it, and we see we get back what we put in. So that is the end of this section. Once again, I'm going to take a quick look to see if there are any questions. Still no questions. All right. So now we're going to move on to the final section, which is good. We only have 13 minutes left. So in this section, I'm once again going to sort of, now instead of just looking at these objects and kind of playing around with them for the sake of it, for instructional purposes, I'm going to sort of go through a somewhat realistic workflow, the sort of workflow that I would go through if I were actually analyzing a data set. So for this example, we'll be using this ALL data set. So what is ALL? So question mark, so it's all caps ALL. If we look at the help section, we see that it's the acute lymphoblastic leukemia data from the Ritz laboratory, right? The data consists of microarrays from 128 different individuals with acute lymphoblastic with ALL. And then they say there are a number of additional covariates that have been added, that have been included. And importantly, this help section contains descriptions of each of these covariates. If you skip to the end, you can see that this is associated with a paper from 2004. So let's just keep this up here. This will be useful. So let's first load the data, and to load it, you just use the data function, data ALL. That's been loaded. Let's start off by taking a look at this object. Now, when we run it, we can see that the output is very similar to what we saw with our summarized experiment object, with our airways object, which we called SE. However, one difference is this is now an expression set object, which again, apologies, there are names like all these funny sounding names that are still floating around. Expression set is a now what's the term? It's deprecated. It's the object type that Bioconductor used to use, and it works very, very similarly to a summarized experiment object. But now, instead of an expression set, people use the summarized experiment object. And that's just because, for example, this expression set object, even though it looks like the object that we were using before, if I now try to run, for example, row data on this, it's not going to work because row data does not work on an expression set object. So what do? I either have to learn about all these new functions associated with an expression set object, or I can just convert it into a summarized experiment object, which is a preferred approach. So it's easy. We can do make. It's maybe my favorite function name because it's ridiculously long. Name of the function is make summarized experiment from expression set. Thankfully, it works super. It's really easy to use. Now, so I've used this function on the ALL object, and I've assigned it to a new object called ALL underscore SE, just to remind myself that the underscore SE means that this is the summarized experiment version. And if we run this, now this looks a lot like our previous SE object. So which means we can now use functions like row data, call data, and so on. Okay, so let's take a look at sample information. How do we do that? We use the call data function. Ooh, sorry, not SE, all SE. We can see, as the help section promised, there's a lot of useful sample metadata. So these are various covariates. We will focus today on this BT column. What's BT? It says here BT contains the type and stage of the disease. So the B indicates B cell, while the ALL, while the T indicates T cell ALL. And if you look here, you can see after the B and T, there are numbers. For the purpose of today, we will just look at the B's and T's without any numbers. So on line 139, I say only keep B and T cells. So how do we do this? We'll have to do some subsetting. So let me create a new object called all underscore BT and subset this all SE. So I want to keep all the rows, right? But I just want to select from the BT column. Sorry. From the BT column, I only want to select the B's and the T's. Now, when I look at ALLBT, the call data for ALLBT, I see in the console there are just B's and T's. So now I have two groups, my B's and my T's. The first group contains 5 samples, and the second group contains 5 samples. So now I can do the simplest kind of differential expression analysis, comparing gene expression between these two groups. So let's proceed. Perform differential expression analysis comparing, comparing B and T groups. Okay, so there are a few steps here. Given that I only have 7 minutes to go, I'm going to go through this pretty quickly. I should say that if you are interested in these details, the previous learning experience that I did, which is recorded, and you can find it, the video, in the on the CCT YouTube page, I go into a lot of detail about how to fit linear models for analyzing different kinds of omics data. So please watch that video. And if you can't find that video, please email me. My email address is my first name dot second name at duke.edu. All right, so we need to do a few things for differential expression analysis. First, so this is microarray data. We need to extract the array data itself. So let's do ALL under, I'll call it LM. So how do we extract the microarray data? We can use the assay function. So assay BT. Right. So the one, I forgot to say this, when you use the assay function, the second argument can either be the name of the assay or the index. So here I'm pulling out the first assay. There is only one assay, so the one's not really needed, but I'm trying to be a little bit thorough. So we have our ALLM, and I'm just going to run it in the console so that I can confirm to myself and to you that this is contains our microarray data, gene expression data. And the second thing I need is I need to produce what's known as a design matrix. And this is where, apologies, I'm going to skip this bit. Actually, before I do that, I need to first create a group. So the group is, I'm comparing the B's with the B group with the T group. So that information is in ALL underscore BT $ BT. But if you were to just run this ALL underscore BT $BT, see, you do get 5 B's and 5 T's. But if you look at the levels, so this is a factor, so it has levels. The levels really should only contain B and T, but it also contains B1, B2, B3. That's because the original data contained B1s, B2s, B3s. I don't want this here because it can mess things up. So what I can do is I can change the levels of this factor to only include 2 levels, B and T. Right? And another reason for doing this is when you're doing differential expression analysis and you're comparing 2 groups, the group that you're comparing to will be the first level. So this way, when I fit my models, the log fold changes or the fold changes that I get will be the fold change of the T group compared to the B group. And if I wanted to do it the other way around, I would set T as my base level. So let me make my design matrix, so model matrix tilde group. So if we take a look at design, this contains information about the 2 coefficients of my model. Again, if this is not making sense, please watch that other video or email me. And I don't like the default names, so I'm going to update the names of this design matrix. Say I only have 4 minutes, which is, I was hoping to be done by now, but we're getting very close. T versus B, looking good. So now I have everything to do my first fit. So you can use the lmfit function from the lemma package. So you put in your microarray data set, and then you put in your design matrix. You run that, and it does it super quickly. And I'll apply this empirical Bayes step really quickly. And now I see if I use the top table function on efit. Let's just look at the first 5 genes. Pull out this T group versus B group comparison. You can see, actually, let me run that again. You can see that I only have the first 5 genes. The most significant gene, meaning the gene that in this case goes up most significantly in the T group compared to the B group, is this gene here. So I have 3 minutes, so I'll add one more step. This is kind of an example of like how I would do a code end-to-end analysis if I was starting off with a summarized experiment object. So I hope you can see that while this might be a kind of strange object to get used to at first, using it to do something useful like differential expression analysis is not especially hard. And everything is in one place. So I'm not referring to different data frames and cluttering my environment. I'm always just referring to this, the same object, the summarized experiment object. I'm going to skip questions. And the final thing I'm going to do, this is using the biomat package, is you can see here that this is information about the probe sets. I have no idea what gene that probe set corresponds to, so I want to look these genes up. Look up these genes. How do I do this? First, let me just save the output on line 153 as ALL LM results. So get the gene symbols. So I'm going to use this function called use dataset. And so the biomat package allows you to query databases that contain all of this rich genomic information. So sorry, I'm rushing. There are two steps. The first two steps of using the biomat package in this case are one, you need to specify what database. And once you specify what database, you need to then specify what dataset you want. So to specify what database, you can use this thing called usemat. Let's just quickly look at this function, the help page. It's the first step in using the biomat package. And you just have to give it the name of the database. And if you want to see what database is available, there's this handy function called listmat or listmats. And the very first database is the one that contains ensemble information for human ensemble information. So we run that. And the second step is to use this function called useDataset, right? And that takes two arguments. That takes the name of the database and the name of the dataset within the database. What datasets are within that database? You can use this function called list datasets and give the mat as the argument. And you can see that there are 214 datasets. So I'm going to scroll up and you can see that there's the name of the dataset and then a description of it. So I know this is human data, so I'm going to look for something that begins with H sapiens. Let's see if I can find it. There it is, H sapiens gene ensemble. So that's the name of the dataset within this database. All right. Now I'm going to use my final function called getBM. Right? Let's pull up the help page so that we can refer to it. GetVM takes in a few different arguments. Let me scroll down. The name of the mat, so that's meaning the name of the data set within the database. This thing called a filter. What does that mean? So the filter is the type of query that you're using. So these are, I mentioned these AFI probe sets. This is where the metadata function comes in use. So if we do metadata on this ALLBT, there's a lot of information here, but at the very bottom, there's the annotation. It gives me the name of the AFI chip, the platform that was used, HGU95AV2. And by pure coincidence, that very same chip was used in this example on the help page. So that's what I'm going to use as my filter, because that's I'm telling Biomat that this is the kind of data that I'm going to be supplying you. I now want you to look up the corresponding, in this case, I'm interested in gene names, the corresponding gene names for these probe sets, the probe sets that are associated with this chip type. So that's my filter. And then I have my attributes. So these are the things I want Biomat to return. I wanted to return, again, just the same probe set IDs, but also the gene names. So you would do external gene name. And finally, I need to give it some values, some inputs. So the values are the probe sets themselves. So to do this, you just do row names, ALL underscore LM underscore results. Why am I doing this? Let's just run row names ALL underscore LM underscore results in the console. And what do we see? We see these 5 probe sets because I'm only looking at the top 5 genes. So when I run all of this, and if we take a look at this AFI to gene, we now see we have a simple dictionary, right? Column 1 has our probe sets, and column 2 has the corresponding gene names. So the final step, sorry, I'm going to skip questions now, just in the interest of time. The final step is I'm going to take my ALL LM results, I'm going to join it with this AFI to gene, right? So the first thing I do is so right now, all the probe sets in this ALL LM results object are in the row names. I want to convert them into a column. Take the row names and put them into a new column. So I can use this handy function called row names to column from the Tibble package. I'm going to give it the same name as the column name as the one in AFI to gene, which is this thing here. Right? I want you to call it this. Ooh, sorry. So if we just run this now, let me clear my console. if we just run this now, you can see what were previously row names now populate a new column called AFI HG U95 AB2. So now we're ready to join. So we can use an inner join, seems reasonable, with the AFI to gene in my console. And we want to be good and specify that we're joining by this common column. So now when we run this, we see that we get our original output, but we also now get the corresponding gene names. So it turns out the gene SH2D1A is the most highly upregulated gene in this analysis. So that is that. I am going to now go ahead and push this to GitHub so you can see all of the code. I encourage you to go through this in your own time. Like I said, please send me any questions you have. I'd be happy to answer them. Thank you for your time and thanks for watching.